In this video, we are going to talk about humoral immunity, how the B cells recognize and respond to an antigen, as well as the actions of antibodies. So with humoral immunity, this is a more indirect method of defense than cellular immunity. Remember, with cellular immunity, we are directly destroying cells that either are infected, that are cancerous, or possibly cells that are foreign, such as bacterial cells. With humoral immunity, this is an extracellular response. So this is not where we already have a cell itself that needs to be destroyed, but where we are trying to limit infection of tissues. So the way that we do this is B lymphocytes produce antibodies. Antibodies are going to bind to antigens and tag them for destruction by other means. So the cellular immunity, once again, attacks enemy cells directly, whereas the antibodies are going to try to limit the activity of antigens until they can be destroyed. The humoral immunity is going to work in 3 stages, just like cellular immunity: the recognition, attack, and memory, or again, the 3 R's: recognize, react, and remember. So for the recognition, we have immunocompetent B cells. These are B cells that are naive still, so they have the ability to respond, but they have not yet been activated. These immunocompetent B cells have thousands of surface receptors for one antigen. So activation is going to begin when an antigen binds to several of these receptors and links them together. This is one of the reasons why very small substances may not be enough to elicit an immune response, because they're not going to be able to link enough receptors. Once we link these receptors together, the B cell is then going to take them in by receptor-mediated endocytosis, which was mentioned in Chapter 3. The B cell digests the antigens once they've been taken inside, and then puts some of those antigens on its own MHC2 proteins so the B cell can act as an antigen-presenting cell. When it displays these on the cell surface, this itself is not enough to cause clonal selection of the B cells. The B cells are going to require the helper T cells to come along. That's going to be important when we talk about HIV in one of the last videos. So the helper T cells are going to bind to that MHC2 protein complex on the B cell, which is now an antigen-presenting cell. Once it recognizes that complex, it will release the interleukins that now activate the B cell and stimulate clonal selection. So now this B cell is going to start to mass produce itself for the response. Most of the cells that are going to be produced in clonal selection are going to differentiate into plasma cells. Plasma cells are larger than the B cells, and they are going to be capable of secreting many antibodies. So they're going to have a large amount of rough endoplasmic reticulum inside of themselves, because rough ER, as we recall, is going to be used for making proteins that are going to be exported out of the cell. These plasma cells can secrete antibodies at a rate of 2,000 molecules per second during their lifespan. Notice their lifespan is brief because these are our effectors. Now, we will also talk about memory cells coming up, and those are the ones that are going to be longer lived. The first exposure to an antigen tends to trigger production of IgM antibodies, so that's going to be part of our primary immune response the first time we see a particular threat. Later exposures are going to result in the production of IgGs, so IgGs are the primary antibody of secondary immune responses. Antibodies are going to be capable of traveling throughout the body, in the blood, and other body fluids. So, when we talk about the attack in humoral immunity, the B cells are not attacking anything, and the antibodies themselves do not destroy anything. What they're going to be doing is they're going to render a particular antigen harmless and tag it for destruction. Now, with the memory aspect, memory in humoral immunity is where we've got some of those B cells that were produced as a result of clonal selection will be memory cells, similar to the T cell memory cells we spoke of before. These are capable of existing in the body for a long period of time. All right, when we look at this picture, what we have is antigen recognition. So in the top part of the picture, you've got the B cells with multiple receptors on their surface. A particular antigen has to bind to multiple receptors, clumping those receptors or binding those receptors together. And then the B cell would actually take those in through endocytosis, digest the antigen, then put that foreign antigen on its surface. And that's where we're now at with step 2, antigen presentation. So that B cell is showing that foreign substance on its surface, on its MHC2 protein. Remember, helper T cells specifically respond to MHC2s. So the helper T cell is going to dock. It's going to recognize that foreign substance, and at that point, it is going to release the chemicals, the interleukins, that officially activate this B cell. Without that helper T cell, the B cell would not have been activated. Again, that's important. So once the B cell is activated, then we go through clonal selection, where we are going to make multiples of that particular specific B cell. Most of them are going to differentiate into plasma cells. The plasma cells are bigger. They've got a lot of endoplasmic reticulum, which is kind of shown here in the pink, kind of the dark pink. They're going to be able to mass produce antibodies that are then going to be put out into the circulation. But then you can also see over to the right, we are going to produce some memory B cells that are going to be in a larger number, and then they are going to be able to linger in the body for years. And now we have that built up immunity where we're going to be able to respond to a threat faster in the future once we've seen it. So this is a nice picture on the left showing a B cell that has not been activated yet. On the right, we are showing a plasma cell, and all those little lines in the plasma cell cytoplasm, that is rough endoplasmic reticulum for mass production of antibodies. So let's talk some more about the antibodies. Again, antibodies are immunoglobulins. We've talked about the 5 classes of antibodies now in previous slides in a previous video. I know that, once again, our slides are a little mixed up compared to what you may have printed out versus how I'm putting them in the videos, but my videos are actually going along with the order that's in your book. So when we look at the function, the action of antibodies, they have 4 mechanisms of attack against antigens: neutralization, complement fixation, agglutination, which is going to seem familiar, and precipitation. So let's look at neutralization first. Neutralization is when antibodies mask the pathogenic region of an antigen. So there may only be one area of the antigen that's actually capable of causing the disease. For example, there may be one area on a virus that is actually the area that the virus needs to be able to get into a host cell. So if the antibodies can attach and cover that up, they have now rendered that virus incapable of getting into a cell. Complement fixation, this is where we are going to have the antibodies attach, and then they may change their shape somewhat and expose areas of themselves that can attach to complement proteins and activate the complement system. So here's where we can activate those different effects of the complement system we talked about with innate immunity: inflammation, phagocytosis, immune clearance, or cytolysis. With agglutination, antibodies can have 2 to 10 binding sites. They can actually bind multiple enemy cells, and then that's going to, first of all, immobilize them from spreading. That's also going to make it easier for them to be removed from the body, because if you've got a phagocytic cell, it's a lot easier to engulf and destroy a group of foreign or potentially threatening cells than to go and hunt them down individually. And then finally, we've got precipitation. So with precipitation, what we are basically doing is we are again binding to the antigens, kind of precipitating them or bringing them out of solution, making them more obvious, clumping them together so that they're bigger and easier to be removed by immune clearance if we're talking about the complement system, or they're easier to be phagocytized by eosinophils. Here we've got an example of agglutination. So hopefully this is tying information together for you from our previous labs, from our previous chapters. We had talked about the red blood cells before. Now we're talking about the immune aspect. But once again, we're seeing our pentamers, right? Our 5-point antibodies here, the IgMs that can have 5 different antibodies attached together. As each of those antibodies can bind to a foreign antigen, then the fact that there are 5 of them helps them clump together, and that's going to make that bigger bite, that bigger chunk of these foreign cells for the phagocytic cells to come and destroy them.